Okay, so today we will be doing chapter 6 of AS level biology. This is nucleic acids and protein synthesis. It's a pretty confusing chapter. A few concepts are very confusing, so I hope I can explain them. So, first we have the nucleotide structure. The nucleotide structure is basically made up of nucleic acids, and nucleic acids include your DNA and RNA, and also ATP molecule we'll learn about further. The nucleotide, the general structure of a nucleotide is that it has a nitrogenous base. It has a nitrogenous base, a phosphate group, and a pentose sugar. Pentose sugar means that it is a 5-carbon sugar. So I'll quickly go through. And then the ATP molecule also has the same thing. So basically a ribose sugar, pentose sugar, nitrogenous base, and the phosphate group. That's basically what you need to learn for an ATP molecule. You don't need to learn like the whole thing like ATP, ADP, and AMP. These are like part of the A2 syllabus, so this is not required. Just know that it has a ribose sugar, a nitrogenous base, and groups instead of one as compared to a DNA or an RNA nucleotide. Then the nitrogenous bases are adenine, guanine, cytosine, and uracil, and also thymine. DNA has adenine, thymine, cytosine, and guanine, and then RNA has adenine, uracil, are paired in a purine and a pyrimidine structure. So purines pair up with a pyrimidine, and then adenine and guanine are your purines. Purines are basically double ring structures, as seen here. We have an adenine structure. So you can see that this has 2 rings right here. So it is a purine. And then pyrimidine includes cytosine, thymine, and uracil, and these are single ring structures. Okay, now we'll look at the DNA structure first. So the DNA structure basically includes a phosphate group and then any nitrogenous base, which is adenine, cytosine, guanine, or thymine. And then the pentose sugar in the DNA nucleotide molecule would be called deoxyribose, as it does not have an OH group, it just has an hydrogen attached over here, so it does not have an oxygen. And it has strands in 5' to 3', and then the antiparallel strand is in 3' to 5' direction. And the bases are paired up like adenine. Adenine is paired with thymine, and then cytosine is paired up with guanine. And this basically, and there's a phosphodiester backbone present as well between each of these bases, which further gives the DNA its helical structure. The phosphodiester bond can be seen here. And then the bases are joined together using hydrogen bonds. Adenine and thymine pair up with 2 hydrogen bonds. This you guys need to learn. So when an adenine will pair up with a thymine, it will form 2 hydrogen bonds. And then when a purine, which is guanine, and a pyrimidine cytosine will pair up, it will form 3 hydrogen bonds. And this is basically known as complementary base pairing, and this is universal. So this is a general rule that you'd have to learn and follow. So if you see over here, thymine and adenine can showing 2 hydrogen bonds, and then cytosine and guanine will show 3 hydrogen bonds. I already told you that it's a double helix structure. That's all you need to know for your DNA structure. And then we have the DNA replication. DNA replicates in a semi-conservative manner. So, it's a semi-conservative DNA replication through which DNA is replicated, which means that one strand, 2 DNA molecules are synthesized, right? So one strand is from your parent DNA, and the other strand would be a newly synthesized strand. So DNA replication occurs in the S phase of mitosis, as we learned in chapter 5. And then the hydrogen, what happens is that first the hydrogen bonds break between the base pairs of the DNA molecule, and this is done by an enzyme known as DNA helicase. The name is not mentioned here, so I'll just write it down. DNA helicase enzyme unzips the DNA molecule, and then each of these single polynucleotide DNA strands act as template for the formation of a new strand. The original strand and the new strand then join together to form a new DNA molecule. So this basically conserves half of the original DNA and makes sure that equal spread of genes occurs. And the whole process is over here. Then another enzyme which will be used in DNA replication is called DNA polymerase. DNA polymerase function is basically when the on the template strand when free activated DNA nucleotides come and bind to the template strand, the DNA polymerase basically catalyzes the formation of phosphodiester bonds between the bases which are joining, and this creates the new strand that synthesizes the new DNA strand, and this forms two templates on the separately on the two template strands. And once all the DNA is synthesized and the phosphodiester bonds are formed, the DNA polymerase just breaks off, and then the complementary base pairs, the hydrogen bonds form between the complementary base pairs of the template and the new DNA strand. Now, there's basically two types of DNA strands in the DNA molecule. So, one is called leading strand and one is called lagging strand. On the leading strand, the DNA is synthesized continuously, which means that the strand is synthesized from a 5' to 3' direction, and using your DNA polymerase enzyme, the phosphodiester bonds are formed continuously and the strand is continuous. But on the lagging strand, the synthesis of the DNA of the phosphodiester bonds is like it occurs in fragments. So what happens is that these fragments are called Okazaki fragments. So these fragments occur because they what do you call on the strand the DNA polymerase moves away from the replication fork, so it just creates like segments or like fragments of these. So another enzyme comes in play over here, which is called the DNA ligase enzyme, which joins these strands and like removes the gaps in between to form another continuous strand to form another DNA molecule. If, as it is seen over here that this is your leading strand, as its continuous replication is happening in 5' 3' direction, but on the lagging being formed, as you can see. So the DNA ligase enzyme will just remove these gaps to create a continuous strand once again. Next, we have RNA structure. So RNA structure is similar to the DNA structure, but we all know that RNA is a single-stranded polynucleotide, and it is made up of, again, many nucleotides linked together in a long chain. Also, like DNA, RNA nucleotides contain nitrogenous bases. So adenine, guanine, and cytosine are the same as such that as in DNA. And then, but unlike DNA, RNA nucleotides never contain the base called thymine. And in place of thymine, there is another nitrogenous base, which is called uracil, present inside this. So the RNA nucleotides also contain another type of sugar, which is called ribose sugar, but this is also the pentose sugar, so it's the same but a different form. So both of these are like shown in comparison. So as I told you that oxygen is present here, so this is your ribose sugar over here in your RNA structure. And then in the DNA structure, you have deoxyribose sugar, so no oxygen is present here. Then this is like a single stranded, and then there's also the phosphodiester present as well. And the sugar phosphate bonds, but the bases are exposed on one side. So an example of an RNA molecule is your messenger RNA, which is the transcript copy of a gene that encodes a specific polypeptide. So other two examples of your RNA are transfer RNA, which is your tRNA, and then ribosomal RNA, which is present in your ribosome. Again, these are for the phosphodiesters formed between 5' to 3' direction. Then we move on to the second part of this chapter, which is your protein synthesis. So how a protein is synthesized from a gene to a polypeptide. So basically a gene is a sequence of nucleotides inside your DNA molecule, which forms your DNA molecule, and this nucleotide basis codes for the production of a specific polypeptide chain. So protein molecules are made up of series of amino acids bonded together, and the shape and behavior of a protein molecule all depends on what the exact sequence of these amino acids will be, which creates the primary structure of the protein. The genes in the DNA molecules therefore control the protein structure, and they determine the exact sequence in which the amino acids will join together to form the primary structure and thus the whole protein molecule. Now, before we head on to the protein synthesis part, we just need to go through this as well, which is the universal genetic code. So, a gene, as I told you, is a sequence of nucleotide bases in the DNA molecule and coding for a specific amino acid. And each sequence of three bases, which is called codons or code, is formed in triplets. So each triplet would code for one type of amino acid, and the triplets of bases are known as codons. And there are 20 different amino acids known which are used in protein synthesis. But the thing is that the DNA code is like degenerate. So, which means there are like four bases. So, there are 64 different possibilities of triplets forming, but we only have 20 amino acids available, which means that the DNA code is basically degenerate. So each triplet can code for the same amino acid as well. So this basically is called degenerate, the DNA code being degenerate. Then we also have start codons and also stop codons, which we will learn later on. And these codons don't code for anything, they just are terminating and starting codons. That's it. And the genetic code is universal. So, like almost every organism uses the same code for like each amino acid. So, if like a specific amino acid, a codon is like coding for a specific amino acid in a human being, the same would be for an animal as well. Next, we have transcription and translation. So we'll first go through transcription first. Transcription happens inside your nucleus of the cell, and parts of the DNA molecule will unwind wherever like the gene that needs to be copied will be unwind or like unzip using again the helicase enzyme. And this exposes the gene that needs to be transcribed. And a complementary copy of the code from a gene is made by a single stranded nucleic acid protein known as mRNA. So what happens is once the gene that needs to be transcribed is exposed, free activated RNA nucleotides will pair up using the complementary base pairing and onto the template strand where the DNA is unzipped. And then the RNA polymerase enzyme will form sugar phosphate or you can say phosphodiester bonds between the RNA nucleotides that are attaching onto the template strand. The stop code this like happens, the mRNA is like synthesized till a stop codon is reached, and once that's reached, the mRNA just breaks off, and then once again the double-stranded DNA molecule reforms when the base pairing with the hydrogen bonds form between the DNA bases. Then the mRNA will just leave the nucleus. Once like it exits the nucleus through the nuclear pore, the mRNA will attach we'll go through translation then, and then what translation includes is this is the stage of protein synthesis which is occurring inside your cytoplasm. So the mRNA molecule will attach to the ribosome, and it attaches to the small subunit of the ribosome, which is an additional point that you can learn. Small subunit of the ribosome. And then in the cytoplasm, there are free molecules of tRNA. And these tRNA molecules have triplets of unpaired bases at one end of the molecule, and then an and its specific amino acid on like the other end of it. So the tRNA molecules have anticodons and the mRNA has codons. So what happens is that since we know that there are at least like 20 different tRNA molecules, each having a specific anticodon. So a tRNA molecule binds with their specific amino acids and then just brings them to the ribosome where the mRNA is already present. Now the anticodons on each of the tRNA molecule pairs with the complementary codons on the mRNA molecule on the ribosome. And at each instance, only two tRNA molecules can fit onto the ribosome, bringing amino acids. And these are like attached side by side. So what happens is on the larger subunit, there is this enzyme called peptidyl transferase, which is not mentioned here again. So the enzyme name is peptidyl transferase. What this does is it catalyzes the formation of peptide bonds in the amino acids to create the polypeptide chain. So this happens like continuously. The ribosome moves along the mRNA, reads the codons, and the tRNA containing the complementary anticodons come and bind, and then the polypeptide chain is synthesized continuously till the stop codon is reached. And the stop codons for the what do you call mRNA are UAG. Then we also have UAA and then UGA. There is a mnemonic you can learn as well. I'll send it later on. It's like you are gone, then you are annoying for UAA, and then UGA is you go away. So it's a good mnemonic to learn because these bases are like the basically codons are required to be learned in the syllabus as well. One more thing that isn't mentioned here, I think, is that the mRNA always has like a start codon. So the start codon is always AUG. Remember that as well. These don't code for anything, they're just terminating and starting sequences that are required in the mRNA molecule. And once the stop codon is reached, the polypeptide chain breaks off and is released to be further synthesized, to be modified actually. Is it shown here? No. Okay, so all of the process is shown here. I think I explained everything. Yeah. Then template and non-template strand, I already told you that the template strand is basically where the mRNA molecule, mRNA is synthesized over, and then the non-template strand is just is not used in the transcription process. We just need to learn that make sure to use the terms template strand and non-template strand because that is important. Next, we have post-transcriptional modification. This happens between transcription and translation. So once the primary transcript is released from transcription, this post-transcriptional modification happens. Why does this happen? Is because eukaryotic genes have both coding sequences and non-coding sequences. Coding sequences are called exons, and these are basically what are eventually translated into amino acids to form the polypeptide chain. But the non-coding sequences are called introns, and these are not translated. So during this process, the introns are removed and the exons remain to be used in translation. So when transcription of a gene occurs, both exons and introns are transcribed from the DNA because the DNA molecule also has it. So these are transcribed as well. So this means that the RNA molecule formed will also contain exons and introns. So the exons are fused together in the process called splicing, which is basically joining together of the exons, and the introns are removed. That's basically what you need to learn for this. And then once the splicing is done and the post transcriptional modification occurs, then the mRNA molecule is ready to be translated and form a polypeptide chain. Next, we have this is the last section. Yeah. So this is gene mutation and their effect on polypeptides. So gene mutation basically means a change in the base sequence of base pairs in the DNA molecule. So mutations occur continuously, but DNA is checked as well, and like DNA molecule once synthesized is also checked. But there can be some mutations which are cannot be completely ignored either. So as the DNA base sequence determines the sequence of amino acids that make a protein, mutation in a gene can sometimes lead to a change in polypeptide that the gene codes for. But most mutations do not alter the polypeptide chain synthesized because we all know that genetic code is degenerate. So what if like there is a mutation happening in one of the bases, but the other like the mutated like gene that's formed is still coding for the same amino acid. So it doesn't really matter. So that type of mutation can be ignored. And there are three types of mutation that happens. One is through insertion of a nucleotide. In this, basically, one of the bases is inserted inside the genetic code. So this creates a knockoff effect. So the whole sequence is changed, and the polypeptide chain which will be synthesized will be different as well, and the function of the protein will also change eventually. Then another would be deletion of nucleotides. This basically includes that a random base is just uh, deleted from the DNA sequence, and this changes the amino acid that would have been coded for. And like an insertion mutation, a deletion mutation will also cause a knockoff effect with by changing the groups of the three base pairs further on in the DNA sequence. So if you look over here, the original gene is coding for tyrosine, serine, and leucine. And then the mutated gene has an insertion of the base guanine. So it is coding for the amino acid coded for tyrosine, arginine, and serine. So it just changed the whole polypeptide. It changed the sequence of the polypeptides as well. So same would happen for deletion. One base will be deleted and then the polypeptides synthesized using that would be different as well. Now then there's another one which is called substitution of nucleotides. This is basically when a DNA sequence is randomly swapped for a different base. But unlike an insertion or deletion, a substitution mutation will only change the amino acid for the triplet. So if we look over here, the base T is substituted for base A. So this will only affect the amino acid serine, which is replaced for arginine. So this does not create a knockoff effect, as you all know. Now, this substitution mutation can take three forms. One is silent mutation. Now, this mutation does not alter the amino acid sequence of the polypeptide because of the genetic code being degenerate. Then we have missense mutations. This mutation alters a single amino acid in the polypeptide chain. So, an example would be sickle cell anemia, which you guys need to know. In a sickle cell anemia, the polypeptide, the amino acid is changed, which causes the structure of the red blood cell to change as well. And then the nonsense mutations are mutations that creates a premature stop codon. So there must be like a stop codon coming in after like 6 amino acids. So if like there's like substitution, and this creates a stop codon at this third amino acid maybe. So that mRNA molecule will be synthesized till the third amino acid sequence, which will cause the polypeptide chain synthesized to shorten as well, or just change the whole structure as well. So it will cause the polypeptide chain produced to be incomplete and therefore affecting the final protein structure and function. So, cystic fibrosis is an example of a disease caused by nonsense mutation, but you don't need to learn this example. It's like not necessary, just learn the different types of mutations that occur. Now, the effect of a gene on polypeptides is that most mutations do not alter the polypeptide, as I told you. However, a small number of mutations code for significantly altered polypeptide with a different shape, such as the main example that we use is sickle cell anemia. And this may affect the ability of the protein to perform its function. So, in case of an enzyme, if a mutation occurs in its polypeptide chain, that's just going to change the shape of the active site, and then it won't be able to perform its function, which would be to hydrolyze or form bonds. Then a structural protein such as collagen might lose its strength as well, because the shape might change due to some mutation or something. So that's all you need to know about chapter 6. If you guys have any questions, you can ask.